For the next 30 minutes or so, what I'm going to do is walk through some non-human forensics applications of this platform, and I think it will help give you a background as to how we started down this road and then realized along the way with some interactions with the FBI and others that the platform that we were developing originally for biological weapon surveillance and for infectious disease detection had real applications in the human forensics setting. So early funding for this work was derived from DARPA, from Department of Defense, for bioweapon surveillance. And the problem there in bioweapon surveillance is how do you know what to look for in the bioweapons regime? You know, we have a short list of pathogens that we're surveying for. If an adversary has a different short list of pathogens that they're cultivating, we're going to have a real problem. We're going to have a very unpleasant surprise. So how do you look for everything all at once, not knowing ahead of time really what you're looking for? So what I'm going to do is walk through kind of this challenge. I'll articulate it a little bit better. Go through our approach for detecting broad-range pathogens. Give you two examples, one in bacterial space, one in viral space, and then talk about the integrated platform, which is common to these applications and kind of segues into the human forensics hardware portion. So if you think about it at a high level, biological weapons defense is not just about anthrax. It's probably on the top of everybody's list, but it's not the only thing on the list. There's a lot of other things on the list. And in different sectors, the same thing holds. If you're interested in food safety, you probably have 0157 on your list of pathogens to look for, but you got a long list of things, and it may be dependent on what sort of foods you're interested in or what geography you're studying. Hospital-associated infections. In the States, probably staph is at the top of the list. In Europe, it's probably Acinetobacter or other pathogens. So the point is that there's a lot of things to detect, be they bacteria, viral, fungal. There's an interesting series of papers by Louise Taylor's group, and they went through and categorized over 1,000 agents known to infect humans. That's at the species level. So there's over 1,000 species of viruses, bacteria, fungi, parasitic protozoa that can infect humans. This does not include at the species level plant and animal pathogens, nor does it include the numerous strain variants. So we know that there are hundreds of different strains of Staph aureus that can infect humans. I'll show you examples in viral space where there's hundreds of viral strains. All are influenza, but different viral strains. And this doesn't include the potential for bioengineered organisms. So that the big picture challenge is, regardless of which of these sectors you're working in or others, how do you look broadly when you're not sure exactly what you're looking for? So if you think about mainstream bioagent detection techniques today, there's still a lot of culture, both in clinics and in surveillance atmospheres. A lot of culture is still done. This detects a subset of all pathogens, and it can take days or weeks, and not everything can be cultured with the right knowing ahead of time which media to use. There's more and more single agent nucleic acid tests coming on board, typically one test per one agent. So in the biodefense arena, you might have a test for smallpox, a test for anthrax, a test for plague, and so on. The problem with this is, if you're doing broad surveillance, be it for bioweapons or clinical diagnostics or food safety or whatever, you need too many tests if you're going to cover everything. You've got to do a lot of one-at-a-time tests. And even if you had a test for every pathogen and you could afford to run a test for every pathogen, you would fail to detect newly emergent pathogens, things for which you haven't seen before, you don't have a test for. You're going to miss them the first time. You're going to miss SARS. You're going to miss West Nile virus. Okay? You're going to miss H5N1. So I show this slide as a prelude to the next slide. This slide shows a bunch of threat symbols and government agencies which worry about these sorts of threats. So there's NIAID category A, B, C priority pathogens. There's threat symbols for animal pathogens, CDC notifiable agents, foodborne, biological weapon, and so on. If we take these threat symbols now and superimpose them on a bacterial tree of life, While you can't probably read the individual entries, the point is going from gram-positive around to gram-negative, there's threat symbols everywhere. There's not just one node of this that we have to worry about from the perspective of biodefense or human health or foodborne pathogens. Everywhere you look, there's something. You'll see things up here that have lots of symbols. These are the brucellas. Bacillus anthracis is down here with a lot of symbols. But there's not just one lobe of this that we need to cover from an infectious disease standpoint. We've got to cover the whole thing. So we do so based on looking at nucleic acids. And there's a couple of important reasons for this. First and foremost, as you know, all living things rely on DNA or RNA or both to propagate. All infectious agents have one or the other. Prions, I don't know if you want to consider those infectious agents or not, but they don't have DNA or RNA. These are unique biomarkers in that they can be amplified. There's a number of ways to amplify these biomarkers, including PCR, but there are other approaches too. We can amplify these from trace amounts of samples, from highly degraded samples, and importantly, from complex backgrounds. This has obvious implications in the forensic setting where we're amplifying human DNA, but also when we're amplifying looking for pathogenic DNA from environmental samples. The fact that we can amplify these and look for these agents without culture is very important. Also, if we're looking at the genetic information, there's more resolving power to be had there. We can look at differences that don't result in phenotypic differences. There are SNPs, as we know. There's VNTRs, there's SNPs in ribosomal RNA that don't result in a different protein being made or protein variants, but we can detect these at the DNA level. And using PCR, we can tune the sort of amplification we do to either be very broad, as I'll show you some very broad applications, or look at genotyping and strain typing, kind of moderate level, or be very specific and ask questions about drug resistance or virulence markers or strain type. So the way we interrogate amplified nucleic acids, obviously many of you have seen a lot of these, a lot of sequencing. We can sequence, arguably the gold standard for DNA interrogation. We can use things like intercalating fluorescent dyes. We can do hybridization with specific probes. DNA microarrays, we can melt and see if there's SNPs present. We've all run a lot of these slab gels, and you've run a lot of these capillary gel electropherograms. As I described earlier, though, what about the mass? There's a lot of information inherent in the mass of a molecule. And as I described previously, from weighing this DNA, we can get a base composition. And that's our signature. That's our human forensic signature. That's our name that bug signature in this application. That's our profile that infectious agent signature. So I'm going to walk through now kind of the Ibis concept for broad range pathogen detection. And you'll see the parallels with this approach versus what we're doing with the human forensics. So here we've got 4 basic steps. These are the 4 same basic steps that we use in the human forensics applications, except for this step is quite different. So step 1 is a primer design step. Obviously for the forensics it's pretty straightforward. We've got STRs, we've got SNPs, we've got mitochondrial regions. We find regions that are highly conserved, that flank a variable region. This is a bioinformatics step. We bring in lots of sequences. We do sequence alignments. We find these regions. I'll show you a couple examples of these. We do PCR. We take those PCR products, and instead of sequencing or instead of hybridizing, we weigh them on a mass spectrometer, and that weight gives us our base composition profile, which in this case can be used to identify a pathogenic organism. So I'm going to go through each of these 4 steps in a bit more detail. Here's an example of a broad-range primer in bacteria. This is derived from a 16S ribosomal subunit. You can see it on the left-hand side of the screen here, a bunch of different types of bacteria, very different types of bacteria. There's E. coli, there's Mycobacterium, there's staph and streps and so on. All of these have this highly conserved region here, which will all be amplified with this primer pair. And over here we have similarly a very highly conserved region couple of variations here which are tolerated by PCR. So this primer pair, this primer plus this primer, will amplify any of these organisms and many, many other hundreds that are not shown on this list. If you look at the variable region here, though, what you'll see is we've got insertions, we've got deletions, we've got SNPs all over the place. So this is highly diverse here, but highly conserved here. So the approach is we're going to take this primer pair and we're going to amplify whatever is in the sample, and then we're going to decode it based on our derived base composition of this variable region. So step one is a bioinformatics step, primer design sort of step. Step two is PCR. We do PCR to give us our broad range coverage. We do cycling to tolerate mismatches on the first few cycles in this case. For the clinical applications, we have an internal calibrant, an internal PCR calibrant for each well. And we do multiplexing. We typically don't multiplex these broad range primers because they're kind of inherently multiplexed already. Any bacteria will give us a signal from that last primer pair. So they're kind of inherently multiplexed. We do more multiplexing with more specific primers where we're looking specifically for strain types, drug resistance, virulence markers, and so on. And obviously in the human forensics assays, we multiplex quite a bit. I described before how we do the electrospray ionization step. That's shown here. Again, importantly, we start with a double-stranded PCR product in solution. Using the appropriate solution and interface conditions, we peel the two strands apart, and we independently measure now in the gas phase with our mass spectrometer the forward and the reverse strand masses. That gives us a molecular weight. Remember our coins and scale analogy. We have masses. We know the mass of the building blocks. We have an excellent scale in our mass spectrometer. Watson-Crick to the rescue. And from this we can derive base compositions unambiguously. Now what do we do with those? Let's think about this in the context of a biological context. So here's an example of a base composition that we've derived, A34, G31, C29, T27. Now we can plot this on a three-dimensional plot. For the time being, just ignore T. So we're going to do 3 dimensions, numbers of A's, number of G's, number of C's. And we can, knowing where we're priming, which genes we're using, we can plot on here, well, where would other bacteria be? Where would we expect to find things? So here's where staph would be. Here's a staph epi. Here's a strep pyogenes. Here's strep gordonii, and so on. So we can bioinformatically populate this with where would things be, and then ask the question, with our measured base composition, with what is it consistent? And in this case, we can see that this base composition is consistent with Bacillus anthracis in this region. It doesn't necessarily mean that this organism is Bacillus anthracis. It means that this region is consistent with Bacillus anthracis. But importantly, it tells us what it's not. It tells us this base composition we've measured is inconsistent with this organism and this organism and this organism and many, many other hundreds of organisms. So by going through multiple regions around the bacterial genome, each base composition can rule in a very small slice of possibilities for an organism ID, and it can rule out a virtual universe of possibilities. So by using multiple primer pairs, we can actually triangulate down and identify an organism of interest. So basically what we're doing is, instead of asking a specific question, instead of asking, is there Staph aureus, is there Bacillus anthracis, is there FT, we're asking what's in the sample, or which pathogens are in the sample. So I'm going to show you a nice example of this, I think, which illustrates looking broadly. This is an example of work we did several years ago now with the Marine Corps Recruit Depot here in San Diego. They had a very virulent outbreak of respiratory pneumonia. It involved 160 recruits being hospitalized. There was 1 fatality. They suspended all training activities on the base. They prophylactically gave everybody antibiotics. There was great concern about this virulent strain leaving the training facility and reaching the civilian population and getting into schools and households. We were sent some samples from Commander Kevin Russell right around Christmas of 2002. We had been running some bioweapon surveillance plates that cover a broad swath of bacteria. So we hijacked some of these plates and looked at some of these clinical specimens. We did some follow-up surveillance at multiple military bases. And importantly, we did some direct analysis of throat swabs without culture. So the first samples that were sent were post-culture. They took throat swabs from sick recruits, cultured them using a strep-biased media, and sent us isolates. And everything we ran had base compositions all consistent with strep pyogenes. Not real surprising. You culture for strep from sick people, you're probably going to get some strep. In all primer pairs, it concluded that this is strep pyogenes. This was post-culture, though. So the next question was, well, what if we look pre-culture? You know, culture can bias what you see. Let's take a raw throat swab from a sick recruit and see what we see. So this is a direct analysis of a throat swab. An individual that had been cultured, we ran it, was positive for strep, but we took a duplicate throat swab and ran it directly without any culture to see what we see. And interestingly, looking at the raw mass spectrum here, we see three sets of base compositions. One consistent with strep pyogenes, that was not surprising. One consistent with Haemophilus influenza, which was surprising. And another consistent with Neisseria meningitidis, also surprising. So we weren't asking specifically, is there H. flu present, is there Neisseria present? We were asking, based on these broad-range primers, what's in the sample? Okay, so this is a 23S broad-range primer pair. Same story with different primer pairs. Here's another broad-range primer, a 16S, same story, strep pyogenes, Neisseria, Haemophilus influenza. These assays, the clinical assays, all have calibrants present, PCR calibrants, so we can actually use the y-axis of the mass spectrometer to back calculate how many genome copies are in each PCR reaction, which is important in clinical applications. So what I like about this example, though, is we really didn't know what we were looking for. We expected there to be strep pyogenes present based on other tests, but using one broad-range assay, we were able to detect three different pathogens, strep pyogenes, Neisseria meningitidis, and Haemophilus influenza. At three very disparate regions of this bacterial tree of life, we did so both quantitatively with about 1.5 million genomes total, and qualitatively, the ratio here is about 4 to 2 to 1 of Haemophilus to Neisseria to strep. Okay, so this is a nice example of really not knowing what you're looking for, but exploiting the conservation of bacteria across ribosomal genes and across other broadly conserved housekeeping genes. We published this in PNAS a few years ago. There's a lot more detail in this paper and a lot more supporting information about other tests we did to convince ourselves and convince the reviewers that what we're seeing with the mass spectrometer was consistent with other gold standard techniques. So let's talk about viruses now. Viruses are a different beast. They don't have a universally conserved genetic element like a ribosome. So there is nothing we can target. You know, viruses can't really agree on their strategy, right? There's no bacterial tree equivalent. There's like a viral shrubbery equivalent or something. You've got RNA viruses. You've got both single-strand and double-strand, positive and negative sense. You've got single-strand DNA viruses, double-strand DNA viruses. They're all over the map. There's clearly no element like a ribosome that are present in all bacteria or present in all ribosomes that we can exploit. So what we have to do then is go through and treat each family as a separate entity and do a bioinformatic study on each family of interest and design primer pairs which will cover all members of that family that are relevant to our assay. So the example I'm going to show you here is that of influenza virus. Okay, so we've put together an assay which covers all members of the influenza family, bioinformatically mining public sequence information. We've got some great collaborators who give us sequence information, and deriving base compositions de novo from specimens that are sent to us. We have a huge database of influenza base compositions. The assay was put together with some great collaborators, which I'll acknowledge here, folks here in San Diego, Naval Health Research Center, Commander Kevin Russell and his group, state departments of health in both Texas and New York, Stan Lemon at the UT Galveston, and a veterinarian, Dave Stallnick, at the University of Georgia. So this is what the assay looks like. This is an 8-primer assay. It's one column on a 96-well plate. We've got primers targeted, one to this PB1 gene, which is present in all flu. All influenza A, flu B, flu C have this gene. I'll show you the alignment. It's nicely conserved across all species. And then we chose other regions on different segments of the virus that are conserved across either flu A or across flu B. So if we have a flu A isolate that's present, we'll get a signal from this PB1 gene, and then these additional 5 primer pairs. If it's flu B, we'll get, again, an amplicon from the universal gene and these 2 genes. And if there's a mixture, we'll get amplicons in all 8 channels, and we'll be able to deconvolve that mixture directly. We do, again, the amplification into the mass spectrometer to base composition. So here's that alignment, the PB1 polymerase. If you look carefully, you'll see down the left-hand side a list of different types of influenza viruses that are very different. There's human and swine up top, kind of the generic garden variety influenzas that we're susceptible to here. There's canine and equine. There's bovine, a bunch of avians down here, and then influenza B and C. Now this list goes down many, many thousands of entries. So there's a lot of viral isolates that we haven't seen in the laboratory before, but we've seen them bioinformatically, and they've been considered in the design of these primer pairs. So in theory, anything on our list of many thousands of entries will be amplified with this primer pair and will give us a base composition, which we can then look in a database and figure out where it came from. Notice also there's a 6 base deletion here between flu A and flu B. So if we plot these base compositions now a little bit differently, where we have numbers of A's, numbers of C's, and then total length on one axis, you can see that a single primer pair, this universal primer pair, will easily separate all flu A from all flu B from all flu C. Okay, so one primer pair giving us a base composition will tell us, is there any flu present, yes or no? And if yes, is it flu A, flu B, or flu C? Okay, but by adding these additional primer pairs, we can get much more highly resolved information. Such as, here's an example of a couple of specimens positive, samples 1 and 3 here. The result is positive for flu A. That base composition, though, is consistent with an H3N2 serotype from New York from 2003. So this gives an exquisitely high resolution look at an infection that really the only other way to do this would be to take the influenza virus and do a full genome sequence. We've done a bunch of preclinical studies with our collaborators. We took a collection of over 600 clinical specimens that were acquired over a 5-year period. Some of these going back to the late 1990s, so some of these samples were kind of aging when we ran them. But importantly, looking at over 600 samples from different sample types, we still got over 97% sensitivity and specificity, and we correctly identified all the influenza A types. There was about 150 H3N2s, 34 H1N1s, and 67 flu Bs. We did see some mixed infections, and here's an example of a specimen that was infected with both influenza A and influenza B. So here's our amplicon peak pair. Remember, Watson-Crick base paired for influenza A. Same primer pair also gave us a pair of peaks consistent with influenza B. And remember, for these assays, we have an internal calibrant, which we can use to judge the y-axis and the y-gain of the assay. We did a lot of validation studies with different types of samples, both blinded clinical samples. We got a bunch of avian influenza isolates, both normal avian influenza and the high pathogenic H5N1 sorts of isolates from all over the world. Importantly, the assay that we run for influenza from birds or from animals is the identical assay to that which we run for humans. Remember the bioinformatic alignment I showed you up front took all those flu sequences into context to make our primers. So essentially any influenza virus should be amplified with these assays and be detected and strain typed. And that's what I'm going to show you here. Without any change to the assay or the primers, basically the difference is on the clinical specimens we're swabbing the throat of a human, and on some of these avian ones we're swabbing the back end, the cloacal opening of a bird. But everything else is the same. Same primers, same software, same hardware, same everything. So we got a set of diverse avian influenza virus isolates from Dave Stalnick at the University of Georgia. These were collected over a 6-year period from different host species, all different funky serotypes, H and N types we'd never seen in the lab before. We'd never seen a H10N7 in the lab before, but we'd seen it bioinformatically, so it was considered in how we designed the primer pairs to cover it. We were sent these isolates. We wanted to test the breadth of our panel, of our flu panel, the same panel we use for humans. How does that work for a true avian panel? And what we saw was we had excellent detection across all primer pairs. All 6 flu A primer pairs gave amplicons in all isolates. We had one, just a blank sample where nothing fired. But all isolates gave base compositions across all 6 primer pairs. You'll notice that some of these primer pairs, these are color-coded, so different color means different base composition for each primer pair. Some of these are extremely diverse. Some of these give us more base composition alleles than we have color in PowerPoint, and some of these gave maybe 2 or 3. These are helpful in more of the human resolution aspects. These are very informative in the avian influenza aspects. But importantly, what was exciting to us is the same panel we've been using for human surveillance, for clinical human specimens, worked exquisitely for these different avian isolates from all different sample types. So I hope I've given you kind of a peek on the other side of how this can be used for biosurveillance, for clinical applications, for food safety, microbial forensics. I hope I've convinced you that just by weighing DNA, we can derive an unambiguous base composition, and that is a remarkably informative signature. We're going to talk more about that from the human perspective throughout today and some tomorrow, with how does that relate to sequencing mitochondrial DNA, how does that relate to sizing STRs with CGE. I've shown you here that using broad-range primers, we can triangulate and identify both bacteria and viruses. The same platform enables us to look at broad bacteria, such as the throat swabs. We did some clinical specimens with human influenza and avian isolates. We've got other assays that we're developing, which I didn't have time to talk about here. And basically what we're asking in diagnostic space is not, is there a specific pathogen in this sample? We're asking broadly, what's in the sample? Okay, that's kind of the take-home point here. So I'm going to go just briefly now into the hardware overview. You're going to get to work with some of this hardware tomorrow. You're going to actually set up samples and actually run samples. A little bit of a history on the system as it is derived. We started this work back around 2000 on what we called the Tiger 1.0, on a system that basically took up 4 rooms worth of equipment. Okay, so the mass spectrometer we used was a very high-end Fourier transform ion cyclotron resonance mass spectrometer. It's like the Cadillac, the Lamborghini of mass spectrometers, which would take up about half of this room. It has a superconducting magnet, a 7 Tesla superconducting magnet, cooled by liquid helium, cooled by liquid nitrogen, vacuum of about 10 to the minus 10 torr, which is lower than a space shuttle orbit kind of vacuum. Very, very high-end. Very, very much overkill for what we're doing. Okay, we don't need 1 part per million. We need about 25 parts per million. We had different rooms for plate setup, for DNA extraction, and for PCR. We built a couple of different incantations of systems with a time of flight mass spectrometer on the back end. we deployed several of these, the so-called Tiger 2s, to government labs. We had an integrated deck that did sample extraction using QIAGEN and plate setup. And then on this deck, it did PCR with purification and mass spectrometry. We've changed some years ago to what's now called the Ibis T5000 platform. This is what you're going to be working on tomorrow. It basically takes the front-end workflow offline from this and starts with amplified PCR plates. And we're working now closely with our new parent company, Abbott, on the next generation system, the Ibis T6000, which is really built from the bottom up with highly regulated assays in mind, both clinical and forensics assays, with all the appropriate design control and quality metrics required to run these assays. So we switched instrument platforms from the big A deck and big B deck based on some concerns about footprint and cost. So we're working closely with folks at Hopkins and CDC, and the feedback kept coming back. This is great technology, but we don't have the space for these two honkin' big decks. And we've already got DNA isolation. We've already got PCR. We've already got robots to set up plates. Why can't we use those and just use your back end? And that was really the genesis of what's now called the T5000. We have many of these deployed around the country now, several now at the FBI lab in Quantico and the DNA 2 unit as well. In their unit, in the DNA 2 unit, they're using the T5000 doing primarily mitochondrial DNA forensics with the goal of replacing existing sequence-based methods. You're going to hear a lot more about this later today and tomorrow. I think it has real advantages in cost, throughput, looking at heteroplasmy and dealing with mixtures. And we're working closely with the FBI now to finish validation of this assay in their hands. This is the hardware which they're validating and which you're going to work on tomorrow. This is a T5000. Basically what it does is takes amplified DNA products, it purifies them, it removes all the salts and DNTPs and polymers that prohibit mass spectrometry. It's fully automated. The robotic arm moves plates around from the desalting module to a heat sealer to the mass spectrometer. Other functions are performed offline, and Kristen Lowry is going to walk you through the whole workflow from extraction to amplification to plate setup. And it can do so with very high throughput here. So this does about 1 well per minute. It's about 46 seconds of actually collecting mass spectra and about 14 seconds of rinsing in between samples and waiting for the spray to equilibrate and so on. You'll notice it's based on primarily third-party components. This instrument uses a commercially available mass spectrometer, a thermo CRS robotic arm to move plates back and forth. We've got 2 LEAP auto samplers, one used as it was intended for directly injecting into a mass spectrometer. The other one we've hijacked and built a custom fluidics manifold, and it's on this module that we effect the desalting and the cleanup protocol. We use magnetic bead chemistry to desalt these oligos. All the plates, of course, have barcodes and are barcode read and tracked all the way through the system. And of course, there's computers both to control the hardware and to do the data processing. Now moving forward with Abbott, we're very excited about the next generation platform that's commercially available later this year, the Ibis T6000. Key features are it removes some of the dependence on these complex, high-cost third-party components. There's no robotic arm and there's no heat sealer and so on. It is still compatible with the existing PCR plates and existing cleanup chemistry, so all the validation that's gone on the T5000 is relevant for the T6000. It's the same chemistries exactly that we're using. It's had a bottom-up design with the in vitro diagnostics market in mind. So a lot more attention is paid to having a quality system, having a design history file, quality manufacturing organizations, and so on. This will support 30 seconds per well, so it's got twice the throughput of the T5000, and it will be built by a third party that's an FDA-compliant clinical manufacturing device company. This is what it looks like. It's got a touch screen for operation. It's got reagent vials over here that are illuminated from the bottom with LEDs, so you can just glance at it and see if your liquid levels are okay. Samples go here on a stacker. You can put 15 plates at a time. There's a position for a stat sample. where if you're running, let's say you're doing a databasing project, you got convicted offender samples and you just got millions of them. You're set up with running 15 plates. You've already loaded that, but then you've got a stat sample. You want a sample you want to run right away. There's a drawer here you can pull out, put your sample on, push the drawer in. That sample will get priority and get run next, get run through the system, and then you go back to your normal workflow. So you're going to get a lot more intimate with the T5000 later today on a tour, and then tomorrow with some hands-on applications. So I'll conclude here, try to keep us somewhat on schedule. Thanks.